Hello, my name is Dr. Roderick L. Roll, and today I will be talking about Mendel and the two-factor genetics. We just finished talking about Mendel and the single-factor genetics. We talked explicitly about complete dominance. Now we're going to look at incomplete dominance, codominance, pleiotropy, multiple alleles. Then we'll talk about two-factor genetics, and we'll look at two major concepts, epistasis and polygenic inheritance. 3 degrees of dominance exist in genetics. They are complete dominance, incomplete dominance, and codominance. Performing a pedigree is often important because having a recessive or dominant allele can cause a widespread spectrum of different diseases. When performing complete dominant genetic crosses, dominant and recessive traits are very apparent, as seen in this pedigree below. The first genetic recessive disorder we would talk about is cystic fibrosis. If the individual has the disease, they will have two lowercase c's or two recessive alleles. Now, if they have cystic fibrosis, they're going to have this thick, sticky mucus that is going to block their airway and make it very difficult for them to breathe. So cystic fibrosis is an example of a recessive genetic disorder that follows the rules of complete dominance. It affects 1 out of 2,500 people of European descent. If you have one big C, the big C is going to overshadow the little c. So in order to phenotypically express this disease, you must have little c, little c. Another genetic recessive disorder is Tay-Sachs. If you have Tay-Sachs, we would write your genotype as little t, little t. If you have Tay-Sachs, you're probably of Jewish descent. It affects 1 out of 3,600 individuals of Jewish descent. This disease is going to affect the central nervous system. The third genetic recessive disorder is sickle cell disease. If you express this disease, you will be little s, little s. This will affect 1 out of 400 people of African descent. Carriers of sickle cell disease have one copy of the recessive allele, and they will not typically exhibit the symptoms. That individual's genotype would be big S, little s. In the slide that is provided, the sickled shaped cells are the damaged red blood cells that are in this blood serum. So a carrier would be a heterozygous individual. Other carriers of recessive genetic disorders do not exhibit symptoms either. In this cross, we're looking at two individuals that phenotypically are normal. They are called carriers. They have sex, and they will produce offspring. The probability in their offspring is 1 infected individual, 2 carriers, and 1 individual that is phenotypically normal and genotypically normal. So in reference to talking about recessive genetic disorders, we will discuss phenotypically normal versus genotypically normal. So in order to be genotypically normal, you cannot have a little c. In order to have the full-blown disease, genotypically, you have to be little c, little c. If you have at least one c, little c, you are considered a carrier. So you are not genotypically normal. Another recessive genetic disorder is albinism. Albinism is little to no production of the pigment melanin. Can this be true? Please explain your answer. There was a news article that stated that 2 black parents had a white baby. Think about what we have just covered and answer this question. If you are not complete, please push pause. The parents in this news article were recessive for albinism. I highlighted the cross with a star. They were both heterozygotes. They had one genotypically normal individual, 2 out of 4 not normal, and 1 that had albinism. So phenotypically, 3 out of 4 are normal and 1/4 is abnormal. So these 2 parents had a 1/4 chance of producing this abnormal individual. Other examples of recessive genetic disorders are as followed. Now we will talk about dominant genetic disorders. One classical example is Huntington's disease. Genotypically, this individual will have to have 2 big H's. Huntington's disease affects 1 out of 10,000 individuals. This disorder is not lethal until the person reaches the age of 35 to 45. So in this cross, you have an individual with Huntington's disease having sex with an individual that does not have Huntington's disease. This is the first cross. The probability of their offspring is 50% of their offspring will have Huntington's disease and 50% will not have Huntington's disease. Now, how is this relevant? When the individual reaches 35, they're no longer going to be with us. They're going to die, and then you're going to be stuck raising the individual by yourself. The second cross, we have 2 individuals that are normal. And with this cross, you have a 1/4 chance of producing an individual with Huntington's disease. Another dominant genetic disorder is achondroplasia. Achondroplasia is a form of dwarfism. To be a dwarf, your genome would consist of a big D and a little d. So to be a dwarf, you have to have at least 1 big D. Now, if you have 2 big Ds, that is lethal, and you will not make it to birth. You would die sometime within the first, second, or third trimester. So everybody who is regular height is normal. Therefore, they will be homozygous recessive which they will have two little d's. I would like for you to answer this question. Can a married couple who are dwarfs give birth to a normal individual? If this is true, I would like for you to give me the probability. I would like to see a Punnett square or a branch diagram to illustrate that you know what this question is asking. If you have not completed this question, please push pause. This is how you should set up the cross. Two dwarfs would have to be heterozygote for the individual trait because it is a dominant genetic disorder. So you have to eliminate one of the possibilities in the offspring because that offspring would die during gestation. So if we eliminate the first square that has the big red X, you only have 3 boxes left. So the probability of these two dwarfs producing a tall individual or a normal individual is 1/3. And 2/3 probability to produce dwarfs. What will a cross between a dwarf and a normal individual produce? What are the genotypes and phenotypes of the offspring? Please write out your data. Write out the cross if you do a Punnett, or write out a branch diagram if you do that way. But please don't just write something about seeing something on a TV show. I would like for you to prove that this can or cannot happen. If you have not completed this cross, please push pause. A dwarf meets a normal individual and they create babies. The probability that they produce a normal babies is 50%. Other examples of dominant genetic disorders are as followed. Most traits occur in a range and do not follow simple, complete, and recessive patterns. This was seen in Mendelian genetics when we were dealing with simple plants like a pea plant. Incomplete dominance is when neither allele is dominant. During incomplete dominance, the F1 hybrid has an appearance that is between phenotypes of the two parents. In this cross we're looking at, we have a purebred red flower crossed with a purebred white flower. All of the offspring do not look like either parent. They're pink. You will have homework questions. Whenever you run across an incomplete dominant genetic cross, please use these big letters with superscripts. They don't have to be the C's with the R's, but it must be a uppercase letter with a uppercase letter that is in the superscript location. So let's look at the cross. A red snapdragon is crossed with a white snapdragon. And all of the offspring will be pink. Subsequently, if you cross two pink snapdragons In the F2 generation, you would get 1 red, 2 pink, and 1 white. The last degree of dominance that we will discuss is codominance. This is when two alleles both affect the phenotype in separate, distinguishable ways. The example we will talk about is blood typing. Now, there's many ways to type blood. You probably heard of the ABO blood typing system. You might have heard of the RH positive versus RH negative. Now I'm going to introduce you to a third way of characterizing blood. It's called the MN blood group. The MN blood group is an example of codominance. If we look at the red blood cells membrane in the picture provided below, you notice that you have a glycophorin A protein and a glycophorin B protein. An individual can express just glycophorin A, they can express just glycophorin B, or they can express both. And each protein is going to affect the body in its own distinguishable way. So both proteins affect blood in separate ways. Look at how we write it out in the slide image provided below. If a person has glycophorin A only, we'll give them a uppercase L with a superscript big M. If they express only glycophorin B, then we'll give them a big L with a superscript N. If they express both of these proteins in separate distinguishable ways, we must give them both letters. Another example of codominance is in reference to cattle. When you mix this red cow with this white cow, you get this roan cow. We're now looking at another cross. We're going to cross a red hair cow with a roan cow. The red hair is going to be expressed on a cell in its own separate way, and the white hair is going to be expressed on a cell in its own separate way. So in this cross, we're mating a red cow with a roan cow. And you will get 50% red and 50% roan. Here is a concept map or a branch diagram of what we've been talking about. We have 3 different types of inheritance patterns or degrees of dominance. The first one is complete dominance. We understand that. The second 2 that we've been talking about are codominance and incomplete dominance. Heterozygotes do not look like one of the parents in both codominance and incomplete dominance. In reference to incomplete dominance, we will produce a blend. The snapdragon will be pink. In codominance, you do not produce a blend. You're going to produce a red hair and a white hair to create a roan cow. Here's another way to compare it using flowers. Think of the cells as being filled with either a red pigment or being filled with a white pigment. In incomplete dominance, the pink snapdragon has half of the cell filled with the red pigment and half of the cell filled with the white pigment. This creates the blend. It's going to be a pink snapdragon or a pink cell. In reference to codominance, you have a red cell that's going to be produced, and you have a white cell that is going to be produced. Now we will talk about pleiotropy and multiple alleles. When one gene controls multiple phenotypical characteristics, this is referred to as pleiotropy. We talked about a recessive genetic disorder earlier, and it was called sickle cell. This occurs when one gene is mutated. It creates a variation in the protein, and this protein will collapse and make the cell collapse to form a sickled shaped cell. But that one variation in the gene is going to cause multiple issues in the patient. They can experience blindness, they can experience liver failure, they can experience a heart attack. So these are multiple traits or multiple characteristics that are caused by one gene. 5 phenotypical characteristics have been observed in individuals with this mutation of the sickle cell gene. Number 1, clump and clogged vessels. Number 2, physical weakness. Number 3, pain. Number 4, organ damage. And number 5, paralysis. Another example of pleiotropy is seen with the pea plant that we have been looking at in Mendelian genetics. This one gene controls flower and the seed covering color. Now we will talk about multiple alleles. In the pea experiment, we had one gene, 2 alleles. But most genes, especially in humans, have multiple alleles. One example is with blood typing. The ABO blood typing has 3 alleles. Big I superscript A and big I superscript B are codominant to each other. And the little I is recessive. So that one gene has 3 alleles. In reference to multiple alleles, if you have the allele big I with the superscript A, you will produce a sugar on your red blood cell that is labeled A. If you have type B, you produce a sugar on the red blood cell that's labeled B. If you have O blood, you don't produce any sugars that are located on the cell membrane of the red blood cell. So this is how we create the blood types. If you have A, you can either have 2 dominant big A's, or you can have 1 dominant big A with the recessive little I. If you have B blood, you can have either 2 dominant big B's or a big B with a recessive I, small I. If you have AB blood, you have 1 of each. Remember, codominance is when these alleles are going to be expressed in their own separate distinguishable ways. And then if you have blood type O, you don't produce any of those sugars at all. So 3 alleles exist in the determination of the ABO blood groups. We use these letters. And like I stated before, The two big eyes are codominant to one another. Now we will discuss two-factor genetics. When looking at two genes, we might be exposed to epistasis. Epistasis is when one gene at one location is going to mask the effect somehow of the second gene at a different location. So we have a classical example of these rats being crossed. We have two black rats that are going to be crossed, but we have two genes that are being transferred to their offspring. If you have at least one big B, you're black. If you have two little b's, you're brown. But there's a second gene that we have to consider. If you have at least one big C, you will express the color. And if you have two little c's, you will not express the color. So if we look in that Punnett square to the far right corner, and we look at that rat and it is white. That white rat is supposed to be brown, but because it has two little c's, the color was silenced. This is epistasis, when one gene at one location silences another gene at another location. The next concept we'll look at in reference to two-factor genetics is polygenic inheritance. Polygenic inheritance is the additive effect of two or more genes on a single phenotype. So previously we talked about pleiotropy. This will be the opposite of that. Polygenic inheritance is when multiple genes affect one characteristic. So at least 3 genes have been shown to affect skin color. So in this example, you have 2 medium colored individuals, and when they have sex, their offspring can fall within these ranges. They can have the whitest of white and the darkest of the dark. Another phenotype that is controlled by multiple genes is eye color. So again, in simple Mendelian genetics, it's one gene, one phenotype. But as you can see, as we move up to talk about humans, it becomes more complicated because one gene doesn't affect one phenotype. It's typically multiple. So in eye color, it is controlled by 2 genes. But at least 13 other genes play a role also in eye color. Height is controlled by over 400 genes. These genes control things such as the long bones, the torso, and the neck. So students always ask about being tall or being short. It is controlled by the length of your femur. Your dad could have had long femurs that he gave to you or did not give to you. Your mom could have had a long tibia that she gave to you. Your grandma could have gave your dad the gene for having a long arm versus a long torso versus a long neck. So all of these long bones, torso, and neck contribute to height. This is controlled by over 400 genes. Weight is another one. And breast size. They're both controlled by many genes. So someone always asks about what does the environment mean? The environment means what you were brought up eating. If you were brought up eating salads as a little baby, you probably still eat salads. If you were brought up eating smoked sausage, smothered pork chops, chitterlings, hot water cornbread, if you were brought up eating that, you're probably still eating that. So that also controls weight and breast size. So it's not just genes in reference to these two, it's genes and what we eat. Genetic testing allows expecting parents to test for possibilities of errors in their unborn child. One genetic test that you can perform is an amniocentesis. This can be performed between weeks 14 and 20. This is when a needle is stuck through the abdomen or through the pelvic area, and amniotic fluid will be collected. In this fluid, you're going to have individual fetal cells that have sloughed off of the fetus. You are trying to capture those cells. Once we have captured those cells, we can now test the fluid and perform biochemical tests and genetic tests. And with the cells, we can perform a karyotype. Remember, the karyotype is nothing but an orderly display of the chromosomes. What you're trying to do is see if you have any abnormal count. If you have any 3 chromosomes or just 1. Remember, you're supposed to have 2 of every pair. So you have 23 chromosomes in a human, 23 pairs. You should have 2 of each. If you have 1, then something's wrong with that embryo. If you have 3, then there's something wrong with the embryo. Then you can act accordingly. In this example, the Phenylketonuria test is a biochemical test that looks for individuals who cannot metabolize the amino acid phenylalanine. So one drunken night, you have an accident. The guy or gal disappears. You want to make sure you get tested if you're pregnant. If you determine that the individual that is developing inside of you has this phenylalanine issue, then if you give phenylalanine in the diet to this individual, you're going to make that developing baby retarded. So performing these tests are vital to some parents sometimes, or some individuals sometimes. The byproduct of the phenylalanine being metabolized is toxic to this individual that have this issue. This can make them become mentally retarded. But if detected early, the newborn can be placed on a special diet that is low in phenylalanine. Another genetic test that can be performed is the chorionic villus sampling test. This can be done between week 10 and 12. A tube will be inserted in through the vagina and tissue will be captured. This placental tissue has cells derived from the fetus. The advantage over the amniotic sampling is that a karyotype can be performed immediately. With the amniotic sampling, you have to take the cells and culture the cells. We have to feed the cells. The cells would have to go through mitotic divisions. Then once we have reproduced enough cells, then they can perform a karyotype. So with this CVS, you don't have to go through that. The cells that were captured from the placental tissue can immediately be enlarged, stained, and a picture can be taken to perform a karyotype. This compares the two. The amniocentesis is between 14 to 16 weeks, and the chorionic villus sampling can be performed between week 9 and 14. So performing these tests is very vital to some expecting parents. But I would also want to warn you about medical errors. According to Johns Hopkins University, medical errors is the third leading cause of death per year. It's followed right behind heart disease and cancer. So what this means is, every time you go into the hospital, you risk dying. This is a review. Know what each one of these terms mean and how they relate to the other.